Thank you, Martin, and thanks everyone for joining today. As Martin said, I'm gonna talk about today the role of circulating microRNAs in germ cell tumors. These are my disclosures, and I'll start with a clinical case just to give you some clinical context of the utility of microRNAs in germ cell tumors. So this was a patient with stage 1 seminoma. So it was a 40 years old man, was diagnosed in December 2017, treated with left radical orchiectomy, had a quite big primary tumor, 9 centimeter classic seminoma, and slightly elevated tumor markers, LDH, just a little bit above the normal limit, and also alpha fetoprotein, which usually it's not associated with seminoma, so probably was not specific for seminoma. And interestingly, the pre-orchiectomy CT scan showed these retroperitoneal nodes. So exactly in the landing zone, he had a left testicular cancer. And was essentially stage 2A because the biggest lymph node was 13 millimeters in the shorter diameter. So after the orchiectomy, the tumor markers normalized, and we asked for a post-orchiectomy follow-up CT scan just to monitor that retroperitoneal lymph nodes. And as you can see here, the lymph nodes had grown. So the biggest one was bigger, measuring 20 millimeter now in the longest diameters and 13 millimeters. There were these two lymph nodes that were very small in the previous CT scan, also enlarging. You can see here better. So he had the stage 2A seminoma, tumor markers negative. And he returned to us. He was completely asymptomatic. Tumor markers continued to be negative, and he did not want to start any treatment without having a confirmation of or a better understanding of his disease. So there are several options here. Chemo and radiation therapy are the standard of care, but at that time we also had a surgical trial open. Or the other option was to repeat the CT scan, although also already the CT scan showed some growth, and then some other option. And I return to this case at the end of my talk to give you context of the importance of new biomarkers in these patients. So this is the agenda of my talk. I'll just give some very short information about epidemiology and some clinical data about testicular cancer, and then microRNAs in testicular cancer, and I'll talk about S1823 and the other clinical trials that are ongoing to validate these microRNAs. And then I'll briefly mention some work we have done into the teratoma biomarkers. So I think most of this audience is familiar with the epidemiology of testicular cancer. So it's an uncommon cancer. It's not rare, it's uncommon, but it's the most common solid tumor in men, in young men of age between 15 and 35 years old. So because it does affect very young people, it's an important cancer. And the incidence is historically very high in Caucasian men, and it's higher in Caucasian men compared to Black men, as you can see in this graph on the left. And the incidence is worldwide increasing, but what we are observing lately is that the incidence is increasing very, very rapidly in the Hispanic population. And we in fact predicted by 2030, the incidence in the Hispanic population will actually become even higher than the historically very high incidence in the Caucasian men. So we don't know really what's the reason for that, but we interestingly observe this kind of phenomenon. There is a very unique aspect of testicular cancer and management of these patients. It's extremely rewarding for medical oncologists because it's one of the few solid malignancies that actually can be cured with chemotherapy, even when the tumor is advanced and it's metastatic. And the reason of this success is related to cisplatin introduction. This chemotherapy drug has in fact changed the natural history of patients with metastatic germ cell tumors. So as you can see here, this is the survival of patients with metastatic disease prior to the introduction of cisplatin-based chemotherapy in patients with germ cell tumors. So most of those patients were dying with no cisplatin-containing regimen. When cisplatin was firstly introduced with PVB by Dr. Einhorn in the late 70s, the survival had jumped to about 80%, and with BEP, which is the current standard of care, it was further improved. So we can nowadays say that most of our patients' metastatic disease can be cured with chemotherapy containing cisplatin. And this is a very young Dr. Einhorn in 1977 when he did the first phase 2 clinical trial in patients with metastatic germ cell tumor with PVB, and then subsequently BEP. And this graph just shows the probability of patients with metastatic disease to survive with chemotherapy since 1977. So we had like a huge impact, obviously, as I show you, on survival. And then like the research in terms of in germ cell tumors really didn't really focus on treatment and discover new drugs, because there was already a cure, but was mainly focused on treatment optimization. So there was a huge body of work that was done to integrate chemotherapy with other treatments. So we did understand the extreme importance of multidisciplinary treatment in these patients. And we optimized the number of cycles of chemotherapy. And there was also some de-escalation of treatment in patients with stage 1, where surveillance was preferred to active treatment. So it was probably one of the few examples of non-risk adapted treatment for patients with stage 1. And more we focus on the quality of life of these patients, because the problem you have when you have very young patients population is the germ cell tumors in a disease that can be cured with chemotherapy. It's to ensure that the patients are cured, so that the treatment is delivered in the right way that can ensure to reach the goal of cure. But you also have another problem. It's a good problem to have, actually, that is to reduce as much as possible, without jeopardizing the cure rate, the maintenance of quality of life for these patients. So in other words, we want to treat only the patients who need chemotherapy. So both over-treatment and under-treatment are equally deleterious in these patients, because even if you over-treat the patients, you are exposing those patients, sometimes without any good reason, to long-term side effects of chemotherapy, which is very concerning in this patient's population, extremely young. So what do we have in terms of biomarker in germ cell tumors? We do have tumor markers. The classic tumor markers are extremely important. This tumor for diagnosis, prognosis, and for the evaluation of treatment response, but only if they are expressed. So the sensitivity is around 60%, and really depends on the histology. So it's higher in non-seminoma compared to seminoma patients. And among the 3 tumor markers that we use, LDH, alpha-fetoprotein, and beta-HCG, the LDH is the one that has the lowest specificity. And we really don't like to use LDH only as a factor to decide about treatment. And this table essentially summarized all the conditions where you can have false positive alpha fetoprotein, false positive elevated beta HCG. This is extremely important to know because obviously if you don't know that there are these kind of false positive circumstances, you may risk to treat unnecessarily your patients with chemotherapy, again exposing the patient to long-term side effect. So this table summarizes sensitivity and specificity of these three tumor markers in seminoma and non-seminoma. So patients with seminoma have expressed tumor markers, mainly LDH and beta HCG, only in 30 to 40% of the cases, while beta HCG and alpha fetoprotein are highly, like more frequently expressed in non-seminoma, especially choriocarcinoma and yolk sac tumor. But again, the sensitivity is histology and stage dependent. It's around 50 to 60%, even in non-seminoma patients. The other diagnostic tool that we originally use in our clinics is CT scan. So the CT scan is obviously limited by the fact that it's based on size criteria. So we consider positive everything it's more than 1 centimeter in diameter, but we cannot really distinguish with the CT scan whether or not the patient has a tumor or has no tumor. And there are a lot of specific conditions that can induce falsely increased lymph nodes. So even if we reduce the cutoff of significance from 1 centimeter to 7 millimeter, we do increase the sensitivity, but the specificity goes down, and we risk to diagnose more patients with false positive, which again is equally deleterious in this patient's population. And those are the operating characteristics of diagnostic CT scans. Again, sensitivity is around 70%, specificity is around 60%. So do we need more precise biomarkers in patients with testicular cancer? So I would say yes, and there are some specific conditions where the tumor markers and the CT scan are limited and show their limitations. One of these is patients with stage 1 disease, so we don't have any good predictive biomarkers of relapse in these patients, and even in non-seminoma, the LVI positivity only predicts 50% of relapse. So it would be very good to have biomarker that actually predicts with high accuracy the risk of relapse in these patients to select patients for adjuvant treatments. In patients with stage 2A, which is one of the most controversial stages without tumor markers expression, a biomarker of viable germ cell tumor teratoma may be extremely helpful to identify patients for curative treatment, so either chemotherapy, surgery. Also because we know from retrospective surgical series in non-seminoma patients with less than 2 centimeter retroperitoneal nodes that up to 40 to 50% of those patients have no disease, so they are false positive. And another, I think, clinical situations where a biomarker would be extremely useful would be the assessment of post-chemotherapy residual disease to select patients for surgery. So can we do better in terms of biomarkers? In the last 10 years, there have been several studies that have shown that there are some microRNAs in germ cell tumors that are overexpressed and can be served as biomarkers, very specific biomarkers for active germ cell malignancies. So microRNAs are a very small fragment of RNAs, only about 22 nucleotides, and they are produced inside the nucleus, and their production is extremely regulated. They're then exported through an active mechanism through the nuclear membranes in the cytoplasm. They are matured, and the single-strand mature microRNA is the one that is functionally active. So microRNAs usually have negative effect on gene transcription, and in germ cell tumors specifically, the microRNAs that have been associated with active germ cell malignancy can regulate cell cycle, and the target, it's most likely this protein LATS2, which is a negative regulator of cell cycle, although there are not a lot of data about the biological regulation of these protein by these microRNAs. So microRNAs interact with the target messenger RNAs and can negatively impact on gene transcription and especially mRNA translation through the interaction with the target RNA and target messenger RNA cleavage, but also with translation repression and with RNA messenger deadenylation. So those are the three mechanisms through microRNAs regulate gene expression. This heat map is nicely showing the high expression of these two microRNAs clusters, microRNA 371 and microRNAs 302 clusters in active germ cell malignancy. So in both pediatric and adult germ cell tumor tissue, in both seminoma and non-seminoma germ cell tumor, there is high expression of these clusters of microRNAs. And in blue here, as you can see, there is no expression, very low expression of these microRNAs in non-cancer healthy testicular tissue or in teratoma. So both teratoma and non-testicular cancer tissue has very low expression of these microRNAs. It's very highly expressed in seminoma and non-seminoma germ cell tumor tissue. So they are not only highly expressed in tissue, but they are abundantly secreted in the bloodstream of those patients. So these microRNA clusters can be detected in the serum or plasma of patients with active germ cell malignancies. And they are more stable than circulating tumor DNA because they are protected by RNase and protease degradation because they're secreted in a complex with some proteins, for example, AGA2. And it's very interesting to see that their expression is strictly related to the stage of the tumor, so with the tumor burden, and they're extremely abundant in the blood, and we use only 200 microliters of plasma to extract these microRNAs. And the expression is evaluated through a real-time Qpcr. The method and the kit are commercially available, and the cost is extremely contained. It's around $60 per sample. Another very important and relevant clinical characteristics for these microRNAs as biomarkers in germ cell tumor is their short half-life. So the half-life is around 2.5 to 7 hours. And we know from studies conducted in patients with testicular mass prior to and post to the orchiectomy that the microRNA 371 specifically disappears from the bloodstream circulation as soon as 24 hours post the orchiectomy. And this is a huge advantage for us because the tumor markers that we currently use have a very long half-life. The alpha-fetoprotein half-life is 5 to 7 days, and also for beta-HCG is around 1 to 2 days. So you imagine that if you have very high levels to begin with, you have to wait sometimes like even several weeks before you can say that the patient has no residual disease. So this table compared the first retrospective studies that were done in patients with testicular masses prior to the orchiectomy and post orchiectomy to evaluate the expression in the serum of microRNA 371 and 3 more microRNAs of the same cluster. So as you can see here, the the curve of the receiving operating characteristics was extremely high for all of them. And in this table here, you can see that when the microRNA 371 operating characteristics were compared to the all 4 microRNAs tested together, there was not really a huge difference. And this is why microRNA 371 was chosen as leading biomarker to be moved forward in the clinical studies. This table also tells us that most of the published studies are retrospective in nature and were conducted in patients with testicular masses prior to and post chemotherapy, or in patients with metastatic disease. So in patients where there was a very high chance to have actually active germ cell malignancy. In terms of technique, all the publications and the studies that have been published so far have used 3 kind of methods. The Danish group has used these target serum microRNAs, while we and the group from Germany and from Cambridge have used this Chiadin serum plasma extraction kit. So these the target serum microRNA extraction kit is a little bit different from the other 2. So they use essentially these supermagnetic dynabeads conjugated with several microRNAs probes. Then they incubate the beads with the blood, and they are able to detect the microRNAs in the blood circulation with no preamplification needed. And then the beads are eluted, separated, and the microRNAs are eluted from the beads, and they are analyzed with real-time PCR. So most of the studies have used the serum separator tubes and serum microRNAs with direct extraction of the microRNAs from the serum. In this case, you do need the 1 preamplification to increase the signals, and then there is a quantification with Qpcr. What we have done, it's a little bit different from the other groups. So first of all, we use Streck tubes to collect our blood. This may have an effect because it does help to stabilize the small nucleotides in the blood circulation. And we use plasma instead of serum. And then the extraction, preamplification, and Qpcr is the same. Another difference between our data and our methodology and the other methodology so far used is that we don't only are able to quantify the relative expression of the microRNAs in the blood circulation, but we can also have qualitative analysis. So microRNA is either positive or negative, and I'll show you why we can do that. So those are the comparison of the methods that I show. I will not go into the details, but the concerns in order to move forward these biomarkers, especially to design prospective clinical trial for validation, have been quality control. So we now know that we need like several steps and levels of quality control for hemolysis, and housekeeping to reduce the variability, and then the data analysis. So most of these retrospective studies have used a cutoff based on a post hoc analysis and based on high sensitivity. But you cannot really do that if you plan to do a prospective clinical trial, because you have to establish the cutoff prior to. So the qualitative analysis is one of the solutions to overcome this kind of limitation to move forward this biomarker in the clinical practice. So what is the clinical utility of microRNAs in germ cell tumors? This is studies where that was published by the German group, so by Dr. Dickmann in JCO 2019, and this is the first prospective study to validate microRNAs 371 germ cell tumor. Again, like most of those patients, had the microRNA assessed prior to the archaeectomy, and several of these patients had metastatic disease, so microRNA was assessed prior to and post chemotherapy. Only 46 patients in this paper were actually patients with relapsed germ cell tumor and available blood at the time of relapse, which we think it's the most important and controversial group of patients where this biomarker could be important. So in this paper, they were able to prospectively validate the very high accuracy of the microRNA 371 detecting active germ cell malignancy. As you can see here, the AUC of the whole cohort was 0.96. For stage 1 was 0.95, and for patients with metastatic disease was 0.99, so it was even higher, in line with what was previously published about correlation of expression and tumor burden. Based on this study, other studies were done, including this from Aditya Bagrodia in Southwest, at Southwest University, where they evaluated microRNA 371 in the serum of patients prior to the orchiectomy in real world. And again, they were able to confirm the very high expression of these microRNAs in patients with active germ cell tumor prior to the orchiectomy compared to control, and the very high AUC again in this patient. So the next question was like, okay, probably this biomarker is extremely important to predict the presence of active germ cell malignancy in a primary testicular tumor. But can we also adopt this microRNA 371 in the most equivocal situation to inform clinical decision? And this is the rationale and the hypothesis that was behind our JCO paper. It was published at the end of 2019. And in this paper, we analyzed under 32 samples from 111 patients. Most of the patients' samples were extracted from our GU biobank in British Columbia, but we also had some patient samples coming from the University of Utah. And this was a very heterogeneous patient population. We had patients with low risk of harboring germ cell malignancy, mainly stage 1 patients. And then the most informative group was the moderate risk patients, which included patients with stage 1B non-seminoma, or patients with stage 2A and negative tumor markers, so borderline elevated tumor markers. And then the high-risk patients included patients with obvious metastatic disease or testicular mass high tumor markers prior to the orchiectomy. So we used the plasma-based assay with the streak tubes, as I mentioned, and those are the results. So as you can see here, we did a quantitative analysis, and our cutoff was simply 0. So it would, the microRNA 371 in plasma was either expressed or was not. So that's why we were able to do this qualitative analysis where we just separate patients with positive or negative, microRNA 371. And in the whole cohort, included 132 samples, we only had 2 false negative results, and the other was truly positive. And we did not have any false positive microRNA results. And those are the receiving operating characteristic curves, the area, the corresponding area under the curve for microRNA 371 in dark red, compared to beta HCG, LDH, alpha fetoprotein, and CT scan in the whole cohort. So microRNA 371 overperformed the classic tumor markers in the CT scan that are the tools that we currently use in our clinical practice. As I mentioned, we did not have any false positive and only 2 false negative results in our cohort. Therefore, the negative predictive value and the specificity was 100%. And also the sensitivity and the positive predictive value were very high because of the very low number of false negative results, so 96% and 98%. So when we look at the moderate risk group, again, the most controversial, the most, I think, informative of the whole cohort, is, probably you cannot really see the swimming plot, but the red dot is the time where the microRNA 371 was evaluated, and then the the triangle, the yellow triangle is when the relapse was confirmed. So the microRNA 371 was able to anticipate sometimes the relapse, the clinical detectable relapse in these patients with moderate risk. And also in this group, the operating characteristics of microRNA 371 were better than the CT scan and the classic tumor markers, confirming that this biomarker has the potential to help a clinical decision to predict at least active germ cell malignancy, even in patients with moderate risk of harboring active germ cell malignancy. So when we were developing this biomarker, one of the critiques that we received was the reproducibility and the repeatability of the microRNAs to be used as biomarkers, because as I told you, because the quantitative analysis is based on relative expression and not an absolute value, there is high risk of variability in the results among like two different experiments or two different groups. So we did this kind of experiment in the JCO paper, and we repeated the experiment twice. And we also asked an external laboratory to repeat the experiment. So we provide the samples and the protocols, and they repeated our experiment. And the concordance was very high. So we didn't observe this variability that was historically correlated with microRNAs assessment as biomarker. So are those microRNAs useful or potentially useful for equivocal clinical situation? I show you the moderate risk of in our study, but there are other studies that have been published, for example, in the post-chemotherapy residual disease. This is a publication from the group of Dr. Hamilton at Princess Margaret Hospital in Toronto. So they observed and they analyzed microRNA 371 and also 367 in serum of patients with metastatic non-seminoma. So they analyzed 82 patients prior to the chemo, post-chemotherapy, but prior to RPLND, and post-RPLND. All those patients presented with the residual post-chemotherapy disease. And again, like in patients with metastatic disease, the microRNA 371 was higher, and then the levels dropped after chemo and also after RPLND. What's most informative in these graphs, I think it's these ones. So when they analyzed the microRNAs prior to the RPLND but post-chemo and correlated the expression to the pathology finding, there was very low expression of microRNA in patients with benign findings, so fibrosis, necrosis, in patients with teratoma, expectedly so, and the expression was higher in the patients with viable germ cell tumor. So they were able also to confirm the high predictive value for active germ cell malignancy in this population with residual disease. So they inferred that this method could be helpful in patients with 3 centimeters or less or smaller residual post-chemotherapy disease. But I think that there are some limitations in this study. The small number of patients, especially the patients with viable germ cell tumor, only 6 patients in this cohort, the retrospective data, and also the lower specificity in this study, because as they did the quantitative analysis, they adjust the cutoff on the base of the sensitivity, and therefore the specificity was decreased, and then they used this completely arbitrary cutoff of 3 centimeters. So there is still a lot of improvement in the post-chemotherapy situation to define the utility of microRNA 371. In patients with stage 1 and stage 2A, this is a small but important prospective study where 24 patients were with stage 1 or stage 2A were evaluated prior to primary RPLND, and microRNA 371 was expressed in the patients with viable germ cell tumor, even if the lymph node size was smaller than 1 centimeter, and not in teratoma and benign conditions. So this is another example or paper of the very high occurrence of this serum microRNA 371 in patients with early stage disease. And then, like very recently, Dr. Hamilton groups, again from Toronto, published this retrospective series in patients with stage 1 on surveillance. So they analyzed 19 patients with seminoma, 15 patients with non-seminoma who experienced a relapse during surveillance, and the control groups was the non-relapsed patients. So overall, 117, 82 seminoma, and 35 non-seminoma patients. And they used the Loyenga method, so the the magnetic beads conjugated assay for the assessment of the microRNA, and they used serum and not plasma. So for the non-relapsed patients who represent the control, this is the samples were collected 12 and 24 months after the orchiectomy. Didn't really specify how many were collected at 12 months and how many at 24 months. So overall, they had these 34 patients, and most of those patients relapsed with the tumor markers negative disease only in the lymph nodes and good prognosis group. And the median follow-up for the non-relapsed patients was 63 months, so was way behind the expected time to relapse in this patient's population. So what's interesting in this paper, this is the whole cohort, and this is the microRNA 371 after the orchiectomy in relapsers and no-relapsers patients. So there was no difference. While when they assessed the microRNA at the time of the relapse, there was difference in expression. And this is in the subset of patients with seminoma, and this one in the subset of patients with the non-seminoma. So the conclusion was that the microRNA 371 was not able to predict when assessed post-orchiectomy the future relapse, but again with the limitation of retrospective studies, not clearly, not the same type and point in the non-relapser patients, and also small number of patients relapse. So do I think that the role of microRNA 371 to predict relapse is over? I don't think so. So there are some data that support that microRNA 371 is overexpressed in the germ cell neoplasia in situ, so even before the tumor becomes invasive. So biologically, I think that these microRNAs are associated with stem cell futures. So they are very early on expressed during the tumorigenesis process in these patients. And we also have some papers that describe a high serum microRNA 371 in patients with germ cell neoplasia in situ. So we also had some few patients where we observed with stage 1 disease, and for example, these patients had a positive microRNA 3 months after the orchiectomy with a negative CT scan for tumor relapse, and then 9 months later the microRNA was even higher, and there was finally a small retroperitoneal nodes that was suspicious for relapse. So I think that we don't have enough data right now to say that it's not expressed in the post-orchiectomy, it's not predictive. And I do think personally that it's more an issue of the sensitivity of the current technique rather than the absence of these microRNA in the circulation, as Dr. Nichols says. Their Buddhas are not in germ cell tumor biology, but in our methodologies. So I think we have to work to improve these methodologies and the sensitivity to detect even few copies of these microRNAs in the blood circulation. And this is a table that summarizes the most important retrospective and prospective studies in microRNA 371 germ cell tumor. And I won't go into detail, but I want just you to focus in the very consistently high AUC of the rock across the different groups. So different patients population, different methodology. We're the only group who actually did follow-up, longitudinal follow-up in these patients, but very consistent high number for accuracy. There are some limitations. One of the most important is the fact that this microRNA is not expressed by teratoma. So we're working on other markers, and microRNA 375 is one of them. And as I mentioned, the cutoff for significance and the relative expression when we use quantitative over qualitative analysis is another issue, but we can overcome it with qualitative analysis. And the variability that has to be reduced with multiple internal control. So it seems that these microRNAs have most of the characteristics of an ideal biomarker, very sensitive and specific, limited cost, available, non-invasive, and with consistent results. But are we ready to use them in our clinical practice? So the short answer is no. In the pyramid of biomarker validation, we are here. We still miss data about the clinical utility that comes from prospective large studies. And this is what we are doing with the SWOG trial. So S1823, for which I am the co-chair with Dr. Nichols, is the prospective observational cohort study that will assess the microRNA 371 for outcome prediction in patients with germ cell tumor. And this is when we went to Indiana University and got the blessing of Dr. Einhorn. So it was a very important step for us and for our group to have his support. So this trial was designed with Dr. Nichols and Dr. Kohlmansberger. And this is the schema. Every patient with newly diagnosed germ cell tumors, stage 1, stage 2, and metastatic disease are eligible for this trial. We only ask for some extra blood. So blood will be collected at baseline, so in the post-urology period in all the patients with the stage 1 and stage 2A or pre-chemotherapy patients with metastatic disease. And then the patients will follow a schedule for serial blood collection until the relapse. And it's every 3 months for patients with moderate risk and every 6 months for patients with low risk. And then at the time of relapse or for 3 years for the patients who are not going to relapse. And the primary endpoint of the study is to establish the positive predictive value of microRNA 371 in detecting active germ cell malignancy at the time of relapse. But because we will be collecting longitudinal and serial samples from the post-urology time until the relapse, we'll also be able to see whether or not the post-urology microRNA expression is predictive of relapse in these patients. And if it's not with the current methodology, we will have this huge clinically annotated biobank to further improve the technique to assess and evaluate these microRNAs. So the study is open through SWOG in the US, and as of Monday, we have enrolled 52 patients. And it's been also open through the CCTG, so the Canadian Cancer Trial Group. And we also enrolled the first Canadian patient at the end of December 2020. We'll be opening the study in Vancouver, hopefully by the end of January. So in order to move forward this microRNA, there are other studies that have been planned. And this study, or PI, is the CM Daneshman in Los Angeles, will be using microRNA 371 to allocate patients with stage 2A or early stage 2B germ cell tumor on the base of the microRNA to primary RPLNA or to observation. So this will be a very important study to assess the clinical utility to inform of microRNA 371 to inform a clinical decision in this subset of patients. And then this is how I envision the future of germ cell tumor patients. So patients with positive microRNA seminoma, I think the treatment would be chemo or surgery, while the patient is negative surveillance. For non-seminoma, because we have the issue of teratoma, for patients with the positive microRNA 371, chemo plus or minus surgery, but also for patients with negative microRNA, observation or surgery will be the option if we will be able to validate this microRNA. So going back to the first case, the patient did not want to start any treatment until we proved the presence of the tumor. So he was enrolled in Geo Biobank. We did the microRNA, was negative, and then because we cannot use this as biomarker to inform clinical decision, we did the biopsy of the retroperitoneal nodes, and it was not seminoma; it was a follicular lymphoma. So the microRNA 371 was truly negative in these patients because it didn't have relapse of seminoma, but had another cancer. And again, this is the limitation of CT scan that cannot really discriminate in a stage 2A patients between seminoma and no germ cell tumor. I would just mention our work in terms of teratoma biomarkers. There was a paper published in 2018 about microRNA 375 expression in teratoma. So this was done in tissue. And we used our samples and our collaboration with Indiana University and with Rob Hamilton in Toronto. And with the urologist, obviously, at VGH to analyze microRNA 371 and 375 in patients with residual teratoma post-chemotherapy. So we had the discovery cohort and validation cohort where this microRNA 375 in the blood was highly expressed in patients with teratoma compared to patients with seminoma or without active germ cell malignancy. And when we combined the two microRNAs together, the accuracy of predicting teratoma was higher than using microRNA 375 or 371 alone. So the AUC in the validation cohort was 0.77 when the two microRNAs were combined together, and this was published in the European Urology in 2020. So is this enough to use those microRNAs to predict teratoma? I don't think it is enough. So we have to further inquire and explore other biomarkers for teratoma, which it's planned to do, especially I think exosomal microRNAs in teratoma may be extremely helpful to discover new biomarkers. This is my last slide. I don't know if you know Daniel Heiss. He's a breast cancer clinician scientist. He's the one who came up with the Oncotype DX, which is now currently used in patients with breast cancer to decide whether or not the patient needs chemotherapy. So he said that about tumor biomarker is as bad as a bad drug. And I totally agree with him, but I also think that a good biomarker is as good as a good drug, and I think that microRNAs are really potentially useful and can potentially change our clinical practice in germ cell tumors, hopefully with S1823 validating those findings. Thanks to all our collaborators, so our the sponsor agency. The Surgical Process Center, our collaboration presence in Smart Hospital, Italy, and to Christian and Craig, who were excellent mentors and helped me to navigate into germ cell tumor work to plan these important clinical trials. Thank you.